Welcome back, class. So again, just to kind of do a little mini review, when we talk about the idea of muscle contraction, remember you have to have an action potential first. So you're going to have that motion from the motor neuron axon release acetylcholine. And when it releases acetylcholine, it ends up in that little synaptic cleft and it binds onto the receptors of a muscle. When this takes place and that action potential occurs, it then releases calcium from the sarcoplasmic reticulum. And when this occurs, now we're starting here. And we're going to start thinking about what happens actually at this muscle contraction. So here, normally in a relaxed stage, you can see that actin and myosin are not bound together. So actin itself has two components, this troponin and tropomyosin sort of look to it. And what happens here is that when you have calcium be released from the sarcoplasmic reticulum, it now is going to bind onto troponin. And when it binds to troponin, it actually changes this figuration between troponin and tropomyosin. And when it does this, as you can see, it exposes these binding sites. They kind of look like little black-eyed peas, if you will, or little olives when you see them. And when those binding filaments are now exposed, now we can see that the actual myosin heads, that thick filament, can now attach and form what we call a cross bridge. So again, if you allow for calcium to come in, it changes the shape of troponin, therefore it alters tropomyosin, exposes those binding sites, and now our myosin heads can actually fit in there and it can form this cross bridging. Now, what you're also going to see as well is that energy is needed in order to make this happen. And when we think about energy, we think of something called ATP, adenosine triphosphate. This is a high energy molecule that allows us to do work. Now, if we deplete it, it ends up turning it into ADP because adenosine triphosphate is an adenine group with 3 phosphate groups. And again, it's a high energy molecule. If we remove one of those phosphates, we know it as ADP. So as you can see here, again, when we're looking at this myosin head, we can see that there's ADP with a phosphate group. What ends up happening is ADP and phosphate actually release from the myosin. And when this cross bridge pulls, it creates what's known as the power stroke. It actually pulls the thin filament. It pulls on the actin. And when this happens and it releases this ADP and this phosphate group, a new ATP can actually bind onto myosin and it breaks this connection to actin. So when this happens, again, it's actually utilizing ATP. And when we use ATP, we release it and split it. And when it splits, this is what creates this power cocking, if you will. So the myosin heads can then actually sort of cock back a little bit and they can store the energy for the next stroke. And this just continues on and on and on. So what I have for you guys here is I'm going to have you guys watch a little mini video, a little animation of sort of what's going on, just to sort of bring this all together, because otherwise it's a little bit difficult to just see from pictures and illustrations. You kind of need to have the whole sort of thing run together.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
So I hope you guys enjoyed that video. I think that's a pretty useful little piece to be able to see and kind of go over in more detail, because I like the fact that it takes you through the nervous system control. It takes you to the idea of the Z lines. It takes you through the idea of actin and myosin and what's happening at that cross bridging. So now in this figure, it actually gives you the idea of the sliding filament model. So again, the reason that we think about it as sliding filaments is that when that sarcomere, so the Z line to Z line shorten, you actually see the actin and myosin are sliding past each other. And so what you'll notice in figure B is you can see when the muscle is just in a relaxed stage, when it contracts, and then eventually when it is completely at its shortened length and cannot contract any further, that you'll see that those Z lines move closer together, the H zones and those I bands are going to narrow up. And the thick and thin filaments themselves, the actin and myosin, don't actually change in length, but everything is overlapping. So again, you're not actually changing the actin and myosin's length, you're more of just seeing the overlapping take place. All right, so again, just to kind of bring this all back together that you can see that that bound ATP to actin, or to that myosin, is going to actually hydrolyze, and it forms that ADP and that phosphate. But what happens here, again, because of the fact that we have calcium allows for that troponin and tropomyosin complex to alter their shape and allow for those binding sites to take place. So it forms that cross bridge. The myosin cross bridge pulls the actin toward actually the center of the sarcomere. And then again, we can see that that ATP molecule has now been hydrolyzed. And then we release that ADP and that phosphate from myosin so that a new ATP myosin can, or a new ATP molecule can actually bind on there. And when this happens, when you bind a new ATP molecule, it breaks that link between actin and myosin. Now, when that breaking takes place, now you're going to see that that AT molecule is split. And then you'll have that sort of power cock back to the myosin head to allow for that store of energy for the next stroke. And then that myosin cross bridge again goes back to that original position. But this will continue as long as there is calcium and as long as there is ATP. This will continue over and over and over again. The moment you don't have that contraction, or the moment you don't have calcium anymore, you're going to stop this muscle contraction. But as long as there is calcium and as long as there is energy, this will continue to take place. And again, eventually it does have to stop. So to stop it, we use what is known as acetylcholinesterase. So again, acetylcholine was that neurotransmitter that stimulated this action potential. Acetylcholinesterase is an enzyme that breaks that down. And so that nerve impulse or that muscle impulse is going to stop when you don't have acetylcholine bound to those receptors anymore. So when this takes place, now you have a bunch of calcium. That calcium is going to actually be stimulated to go back into the sarcoplasmic reticulum. So the sarcolemma, the muscle fiber, they're going to cease that action potential. It won't be stimulated anymore. Calcium goes back into the sarcoplasmic reticulum. And because you don't have calcium anymore, that troponin and tropomyosin complex go back to their original shape. They cover up those binding sites. Actin and myosin can now not form those cross bridging. And when they can't form that cross bridge, it allows that muscle fiber to relax and go back to its original length. Now, again, I told you that as long as there is calcium and as long as there's ATP, you can have muscle contraction. But keep in mind, ATP is extremely useful and it's essential. But also keep in mind that you can lose energy. And so you have to be constantly making new energy. So when we talk about our energy idea, again, the energy that we have is the capacity to do work. And we think about it from the cellular component of adenosine triphosphate. Adenosine with 3 phosphate groups makes up this very high energy molecule. And unfortunately, we utilize ATP a lot. We have many different mechanisms to kind of renew our energy source, but we're constantly moving, we're constantly talking every time we breathe. Again, a lot of it will go to our, just to other metabolic needs that are around. So we actually don't have a great ATP reserve, but we do have a little bit. And one of our ATP reserves that we talk about is known as creatine phosphate. So imagine, if you will, that your body kind of had its own little rainy day fund. This is what it would be. So when your ATP or ATP levels are low, you end up actually storing what is called creatine phosphate. And when you're low on ATP, we can actually steal this phosphate group from this creatine phosphate and generate some ATP. Now, when your ATP levels are high, we don't necessarily need to be making a lot of ATP then. So we can have this be stored as a rainy day. So what happens here is your ATP molecule will actually lose one of those phosphate groups, and it gives it to creatine to make creatine phosphate. So we have a little rainy day fund. So again, when your ATP levels are high, we can just keep storing up that phosphate and creatine phosphate. And again, we can store this in our skeletal muscle. But basically, when our ATP levels are low, we have a little reserve. Now, remind you, this reserve is very small and it's not going to be a great amount of energy. You get about 4 seconds out of it, but it's better than nothing. So this gives you enough, like little boost, if you will, to sort of generate some ATP. And so that is kind of our reserve that we think about. Now, we have other mechanisms as well to produce ATP. And you've probably have heard of these before, that we talk about like anaerobic glycolysis, and especially those that are really into exercise, you probably know about anaerobic versus aerobic exercise and what to sort of do when. Anaerobic is going to go through a phase where it does not utilize oxygen. So you're going into something called anaerobic glycolysis. And this does work to produce a little bit of ATP, but it doesn't produce a lot. And so when we talk about this phase, we're actually going to spend a good portion of the chapter in the digestive system actually talking about this idea of producing energy. So I'll kind of make a little bit of mention about it now. So in our bodies, again, our goal is to make energy. And so you've probably seen this picture before. C6H12O6, and we see O2. I'm sorry, let me fix this. Plus O2. Plus water. So what this is doing is showing you that if you have a little bit of sugar, aka our glucose molecule, and you use some oxygen, you can perform what is known as respiration. And as respiration continues, as your byproducts, you give off carbon dioxide, you give off water, and we give off energy in the form of ATP. And that's the ultimate goal, is ATP allows us to do work. This is our energy molecule. And so what happens here is we can either go through aerobic or anaerobic phases. Anaerobic means with no oxygen. So make a little note about that. Anaerobic is no oxygen versus aerobic is with oxygen. And like I said, we'll be bringing this back up here in a couple of weeks, but I like to at least make mention of it now so it kind of makes a little bit more sense. But in anaerobic glycolysis, you're going to be breaking down glucose into what is known as pyruvic acid. And so that is a step to know, that glycolysis is the splitting of sugar into what is called pyruvic acid. And when this happens, you do produce a little bit of ATP. But you don't produce a lot. In fact, you only produce 2 molecules of ATP. And unfortunately, what can happen is if you do not have oxygen exposure, you're going to end up forming what is called lactic acid. Now, lactic acid is going to be sort of a conversion from pyruvic acid, and we can end up accumulating this in the body. The great thing about thinking about muscles and thinking about contraction is the fact that we can build up quite a bit of lactic acid. And the only issue that we'll end up seeing is if we start forming this lactic acid in great volume, it can create acidity in our body. There is also evidence to show, and this is kind of a kind of a controversy, if you will, that lactic acid storage or lactic acid buildup leads to muscle fatigue, and therefore you won't have muscle contraction. It can lead to muscle fatigue. It doesn't necessarily lead to where you can't have muscle contraction. So I'll talk more about that here in just a minute. But one thing that happens with anaerobic is that you get this energy pretty quickly. So that's one of the reasons that people do have a mechanism or exercises that are considered to be this anaerobic glycolysis. So if you were doing like a really fast sprint, you would be utilizing anaerobic glycolysis. And again, you split that glucose molecule, you get some ATP, very little, but you are able to produce ATP. The only bad thing is, again, you end up with the development of lactic acid. Versus with aerobic respiration, you have that still that glucose being split to pyruvic acid, but now it goes through a new series within what is called the mitochondria. So if you guys remember, the mitochondria is that powerhouse of the cell. That's the one that helps us to generate ATP. So in our muscle, we actually will also find a great deal of mitochondria because, again, we need that energy production. And this does produce actually quite a lot of ATP. it goes through this, what's known as the Krebs cycle or citric acid cycle. And then it goes through the electron transport chain to get quite a number of ATP molecules in comparison to anaerobic. So this is actually way more useful, and you can have majority of your ATP production happen through aerobic respiration. And whatever you're not using, again, we can keep on reserve in terms of keeping it stored in our muscle, as well as we also do store some extra oxygen in our muscles too. This is what we know as myoglobin. All right, so unfortunately, again, we need oxygen and we do need ATP as well. So during our rest, our respiratory and cardiovascular system really do supply enough oxygen to support us through aerobic respiration. But like I said, if you're an anaerobic, so again, someone is doing a really intense workout or they are doing a sprint, you can't continue to sprint for long periods of time, because what will happen is if you're not utilizing oxygen, you don't have sufficient amount left. And instead of having that pyruvic acid go through that new song and dance to the Krebs cycle and to that citric, or to that Krebs cycle into that electron transport chain, you end up producing high amounts of lactic acid. And so we call this oxygen debt. Oxygen debt is the amount of oxygen actually needed to basically remove lactic acid from your body. And when you remove it, it's actually going to restore the muscle ATP, as well as it also restores your creatine phosphate concentrations as well. So basically what you're doing is you're ridding the body of that lactic acid and you're converting it back to pyruvic acid. If they say you're converting it back to glucose, you're actually converting it to pyruvic acid to be able to run through the aerobic respiration. So you actually need to remove this lactic acid and get your oxygen levels back up. So that way, again, you can start forming ATP and you can get back your creatine phosphate concentrations as well. So this is just a little picture to be able to see. So one thing as well to keep in mind, we had made mention of this in chapter 2 for chemistry, but I want to bring it back to you. So glucose is a carbohydrate, and we can store glucose in our skeletal system as well as in our fat. And we store it via this big macromolecule called glycogen. So when we need our glucose, we can call on glycogen to break it down into glucose. And when we break it down into glucose, again, if it went through this phase of anaerobic, you'd go from pyruvic acid to lactic acid. Now we have this great accumulation of lactic acid. So to remove it, we have to actually replace our bodies with oxygen. And you probably have felt this at one point in time too. If you ever played soccer or were running, and all of a sudden you start to feel fatigue in your quads, and you start to feel like it almost feels like cement a little bit. What you have to then do is you kind of take a break, you stop running, you take maybe a moment to sit down, and you start to feel that sort of go away. You have that lactic acid buildup that we're trying to now rid ourselves for. And if we can remove that lactic acid, then we can convert back and then be able to run through aerobic respiration. So a lot of times when we think about lactic acid formation can actually relate to muscle fatigue. And when we think about muscle fatigue, it's the inability to actually contract muscle. Now, to that being said, it doesn't completely wipe out the muscle. The muscle can still contract to a degree. And in fact, actually lactic acid in some levels is actually used to generate more muscle, because it can lead to a little bit of scarring in the muscle, and it can lead to the muscle getting bigger through this hypertrophy. But it can lead to muscle fatigue. So when we think about muscle fatigue, again, the accumulation of lactic acid is kind of controversial. In fact, actually, we had this discussion last year. This was kind of a big, I wouldn't say big fight, but we had like a major conversation about sort of what to do in this situation, whether lactic acid was considered to be part of the muscle fatigue, if we were going to teach it that way, because many textbooks do, or if we were going to just sort of make mention of it. So again, it's quite controversial with lactic acid if it truly leads to the full muscle fatigue and would lead to not having muscle contraction at all. It's not entirely true, and research is showing that. But there's other reasons that you can have muscle fatigue too. You can have it due to decrease in blood flow. Sometimes it can actually be due to ion imbalances between sodium and potassium. It can also be that you just sort of have a loss of desire as well that can lead to this muscle fatiguing too. So sort of that mind over matter idea. And then again, when we talk about lactic acid accumulation, it can lead to some degree of muscle fatigue. And then when we think about muscle cramping, it's actually different than muscle fatigue. So muscle fatigue is the inability to actually contract the muscle versus a cramp is a sustained contraction. It's involuntary. You have no control over it. If you've ever had a Charlie horse, you know what I'm talking about. It's a sustained involuntary muscle contraction. And this is due to an electrolyte shift. So again, issues with our ion imbalances that are taking place can lead to muscle cramping. And it's a sustained involuntary muscle contraction, like your Charlie horse that you think about. Now, keep in mind too, skeletal muscle helps you to produce heat. So heat is actually a byproduct of the cellular respiration. So again, we use glucose, we use oxygen. As our byproducts, we give off CO2, we give off water, we get energy from it, and we also give off heat as well. So just due to this whole metabolic standpoint, we're going to be generating heat. So muscle cells are a main energy source of body heat. So keeping that in mind, again, your muscles are very important when it comes to generating heat. And more than half of the energy that you actually release from cellular respiration actually becomes heat. That's a big thing to keep in mind. Less than half is actually transferred to ATP. Most of it is going to go to maintaining that heat and having that release of heat. And when we think about the heat production, your blood actually transports heat through your core. We're going to talk more about this when we start talking about blood vessels, that's why there's some that are superficial and deeper, because that blood is actually transporting that heat. But keep in mind, skeletal muscle is a major component of body heat. And you also are going to have to think in mind too, that when we think about muscles moving, remember that it's not really just one skeletal muscle fiber or just one muscle that's truly contracting. You have this contraction that's going to be observed by a single skeletal muscle fiber, and that allows us to see and record changes, but that's not really how it works. The contractions are going to be by certain particular muscle fibers, and we can activate more. But when we think about these responses, again, imagine that we took out one muscle cell and we just started stimulating it to sort of record these links and these changes. So one thing that we're going to see is what's called a threshold stimulus. There has to be some sort of strength to get the muscle fiber to contract. So we call that hitting threshold, if you will. It's the minimum strength needed to get this muscle fiber to contract. And when you've hit that threshold, action potential is generated. It's the all or nothing principle. If you don't hit that threshold, action potential isn't generated. If you do hit it, you will get that action potential, you'll get that calcium release, you'll form that cross bridge. And when that action potential goes from the motor neuron to the muscle fiber, again, it stimulates a muscle impulse. So that's something to keep in mind. It's kind of like an all or nothing. It's got to hit this threshold. If it doesn't hit threshold, you're not going to have that muscle fiber contract. So what we're seeing here is called a twitch. And a twitch is a response of one muscle fiber to one single impulse. And so what you're going to see is that there's a period of where it's stimulated, and then there's this period of contraction that's sort of happening right here. So there's a little gap in this little piece. It's called the latent period. It takes some time to actually get this to go. But once it does go, again, once it sort of hit that threshold, it's going to contract. And then what you're going to notice is that the period of relaxation actually takes a little bit of time. It's not as quick as you would maybe think when we think about this sort of response. It only contracts for a certain moment, but it takes a lot longer to sort of get that response to have that relaxation take place. But basically, when we think about a twitch, again, a response from one fiber, one impulse, again, you're going to see a little bit of a latent period. It takes a little while to get it going. You'll see a quick contraction, and then again, you'll see a little bit of a longer relaxation. And what we have to keep in mind too is that the length of the muscle fiber determines the force. So the length of the muscle before you stimulate it determines the amount of force that can develop. So forces can be different in particular muscles. And so the optimum length is the resting length. If you go from resting length to then having contraction, you'll have the greatest force. And I do want you to know that the optimum starting length is resting length. So not contracted, just resting will develop that greatest force. Stretch muscles are going to have less force because the myosin heads can actually reach the binding site. So overly stretched doesn't do a good thing. Same thing with overly shortened. It doesn't develop as much force because it's actually compressed. So the sarcomeres can't shorten any further. So the optimum length of the muscle fiber to get the greatest amount of force would be at resting. And what ends up happening is we end up developing what is called summation. This is a process by which the muscle fiber twitches actually combine. And what they do is they produce what's known as a sustained contraction. And this leads to what we call either a partial or a complete tectonic contraction from happening. So again, you have these contractions kind of combining and they just lead to this sustained muscle contraction happening. So it doesn't have this contraction and relaxation. It's basically all just one sustained contraction taking place. All right, I'm going to stop the video right now. I want you kind of to go back through those ideas and we will pick up at this motor unit component. Bye guys.